Hey there, ladies and gentlemen, boys and girls. Today we're going to talk a little bit about our first means of transportation across the cell membrane. So remember that cell membrane on the outside of the cell, that phospholipid bilayer. Let's think back. You hopefully have your notebook out. You can flip to that page that we did last time. Look at that membrane, right? Remember that is what is responsible for deciding who goes in and out of the cell. It's very fluid. It's semi-permeable or selectively permeable, which means it allows some things in, some things out. Today we're going to talk about one of the methods on how it lets things out. But before we do that, I like to use this little analogy here. All right? I like to use this analogy here. It's something to kind of think back on. It's something I'll reference again later. All right? So let me explain the scenario here. I have two situations here. I have kind of this ramp, right, where I have a high elevation and I have a low elevation. All right. If I were to take a rock and try and push it down the ramp from high elevation to low elevation, all right, it would take very little to no energy for me to do that. All right. It kind of naturally flows from high to low. It rolls down. Now it's due to gravity, right? Now, at the same time, I could also push it back up the ramp. And I could go from a low elevation to a high elevation. But to do that, for that rock to go from a low elevation to a high elevation, is going to take a lot of energy, in that case, from me. All right? And this is a little bit of how we describe the difference. Sorry, trying to cut off the bell there. All right? This is a little bit of the description on how we use, or what we call energy usage when it comes to cells, this idea. All right? If we're going to go from a high to low energy use, or high to low elevation, or in this case, we're eventually going to talk about concentration. But if we're going from high to low, this requires very little to no energy. This is what we also call passive energy usage. All right, so let's keep that word in mind, passive. Let's keep this idea of high to low. All right, now we could go low to high. Right? We can kind of do the reverse, or go kind of against nature in that aspect. But this is what we would call active energy usage, because we have to put energy in to do this. All right? So passive being little to no extra energy needed. Active, you need energy to do that. Look, he's sweating there. All right? It needs energy to go from low to high. Very little energy to go from high to low. Okay. So this is just one of the things we're going to keep in mind as we go forward for this, is this analogy. I'm going to come back to this one a little bit later. But for right now, let's start talking about our first way that things can go across the cell membrane. All right. So let's kind of recap a little bit of what we have with our actual membrane we have here. All right. So in case here I have the outside of the cell and the inside of the cell. Remember, we are made of, or that cell membrane, that fluid membrane, is made up of phospholipids, where we have that hydrophilic head and the hydrophobic tails. Now remember, those tails are facing towards each other because what they're trying to avoid is that solution on the outside that has water, and on the inside that has water. All right? Remember, that solution, most of the time, that solvent is water. And it's avoiding that water here and the water here. So those tails point opposite. So there's water on either side of the solution. All right? And embedded into that phospholipid bilayer are proteins and cholesterol. The cholesterol give it that extra liquidy, or that kind of a fluid motion. It's like slick, almost a little bit like an oil. All right, and then it's embedded with different types of proteins. Some proteins' jobs are for signaling other cells. Some proteins' job is what we call adhere proteins that kind of connect multicellular cells together. And then the one we're really going to be talking about is the transport proteins, the ones that allow things in and out of the cell. All right. And so today, remember, we're talking about, we have a outside of the cell, inside of the cell. All right. Today we are going to talk about how some things can go in and out of the cell. But today, what we're focusing on today is how solutes can travel in and out of the cell. Go back. Remember, solutes. Think of salt water. The salt is the solute. All right, so we have this solution here of water with solutes in them. All right, and we're going to talk about how they can transport across that cell, or that cell membrane. All right. The way that we're going to do that today, the one we're going to talk about today, is something called diffusion. All right, what we call diffusion across the membrane. All right, there's a couple different types of it. All right, there's a couple things that identify fusion. All right, first of all, fusion always occurs for solutes. All right, again, we're talking about solutes here today. The diffusion of a solute. All right, the diffusion always goes from a high to low concentration. High to low concentrations, which means based on what we just talked about over here on the other side, all right, that means that this is what we call an example of what we call passive. Sometimes what we're going to talk about today is sometimes called passive transport because it's high to low concentration. It doesn't really, or because of that, what defines it, it doesn't really require outside energy, all right? No outside energy required. All right. Now we can break this down even further. All right. We can talk about how there's actually two types of diffusion. Right. Now let's talk about this first one here. So let's do an example here. All right. Let's say, for example, on the outside, I take a deep breath. What I'm breathing in is a bunch of oxygen molecules. All right. I'm bringing a bunch of oxygen. Oxygen is O2. All right, so I'm breathing in, and now my lung cells are experiencing, coming towards them, a high level of oxygen coming through. All right, so now I have a lot of oxygen. Now let's compare that to, say, maybe inside my cells, by comparison, all right, I have very little oxygen. All right, so now we're going to talk about what we call concentration gradient. All right, so we see here, I have my solutes, my oxygen. Now remember, there'll be water in here as well. I have my solutes. All right, right now, the way I have this, outside the cell, I have a high concentration of oxygen molecules compared to a low concentration inside the cell. It's how they compare to each other. By concentration, it means I have a lot of it. A lot more of them here than I do here. All right. So I have a high solute concentration outside the cell. Well, we just said diffusion is going to be going from high to low concentration, which means these molecules here, oxygen is very small. It's only 2 atoms, 2 oxygens together. It is not ionically charged. It's not a charge. There's no ions. And so what it does, because it is small, it is able to actually just fit in between those phospholipids. They actually fit right between them. The water will not. Remember, that is hydrophobic tail, so the water won't. But those solutes, we're going from a high to low concentration. This is what we call simple diffusion. So it's diffusion, meaning it goes from high to low concentration, and it doesn't require inner energy. But this is simple diffusion because, all right, we are using very small molecules. Easy way to tell that, is it just a few atoms? Or, is it CO2? That's a carbon, 2 oxygens. If it's just a handful of atoms, that means, 2, 3, 4 atoms, that's very small. Glucose is made up of 24 atoms. That's a bigger molecule. All right? Small molecules, they're uncharged molecules. Which means that there's no charge on here. When you talk about physical science, you can talk about positive ions and negative ions, anions. anions and cations. All right, they're not charged, and they go through the membrane or through phospholipids. They're able just to go right through. All right. Now that's small and uncharged, all right, which means that there's an opposite to this. All right. In the case here, what we're talking about here, let's say, I do a lot of work out here, and what that's going to do inside my cells, some of my cells, by using a lot of muscle usage, is it's going to build up a large concentration of sodium ions. You can tell it's an ion because it's got that little positive charge. Inside my cell, I'm building up a high concentration of sodium ions compared to outside my cell. All right. So first thing I would have to address is, what direction is the concentration going to go? All right. We have a high concentration of sodium ions here. Concentration, see over, so you go ahead and see. All right. We have a lower concentration out here. All right. We are talking about diffusion, but now there's a problem. These oxygen molecules were able to go through the phospholipids because they were uncharged. But now we have an ion. That's a cation. That means it's positively charged. It cannot just flip through those phospholipids. All right, those positive charges are going to attract to those lipid tails, and it's going to tear apart that cell membrane. And so what it needs to do is it needs a special highway to do that. All right, it needs a special path to do that, and that's where we're going to use these transport proteins. More specifically, these are what we call channel proteins. All right, and it's going to use these channel proteins to go from low concentration to high concentration. All right, not through the membrane, but this time it had to use those proteins. Remember, we got our proteins here. All right, so we have our protein. This type of diffusion is what we call facilitated diffusion. Facilitate means basically with help. Facilitated diffusion. All right, this is either for large or charged particles. Large molecules, charged ions. Again, we can tell what's an ion because if it has this right here, this positive charge, that's telling me that's a cation. All right, that's an ion right there. All right, charged ions, and when it does that, it needs a transport protein to do that, or a channel protein. Channel protein required. All right, so we have two different types of diffusion here. This seems like a lot. There's a lot up here. There's a lot of colors. All right, so let's recap it here. We have that membrane. It was specifically structured to design certain things in and out of the cell. All right, with those phospholipids. Again, remember why the tails are facing. All right, things are going to go in. One way that can happen is something called diffusion. All right, diffusion means it goes from high to low concentration. It's passive, which means it doesn't need any outside energy. All right, now there's two types of diffusion. With small uncharged, they go right through the phospholipids. If they are large or charged ions, they need a channel protein to do that. But either way, doesn't whether it means it goes through the phospholipids or whether it goes through a channel protein, it needs no outside energy and it goes from a high to low concentration. Both of these went from high to low, high to low. All right? Okay, that's our first example here. That's our first example. This is a diffusion. Oh, one last thing. I want to make sure I highlight. I am talking about the diffusion of solutes. All right, one of the later videos we're going to talk about this, the diffusion of the solvent. That's called osmosis. All right, we're going to talk about that in a moment. So right now we're just talking about the solutes. Think of salt and salt water. All right, the solutes in this case here. All right, that's going to be it. There's a lot going on here. Take a moment. Make sure you can, think about what's happening. Think about the questions to think about. You understand what is diffusion, the difference between simple and facilitated diffusion. What do they have in common? What are they different? All right. Other than that, that's going to be it for today. Good luck, and may the science be with you. Thank you, everyone.